My Forensic Podcast. In November 1983, Randolph Arledge was charged with the murder of 21-year-old Carol Elaine Armstrong. Her body was found on a highway near Corsicana, Texas. She was stabbed 48 times and raped. He was convicted on March 27, 1984 to 99 years in prison. However, in 2006, the Innocence Project lawyers requested DNA tests to be done. This included evidence of a hairnet. However, this failed to find Arledge's DNA and found another man's DNA presence, David Sims. Later on, DNA testing on pubic hair from the victim confirmed that David Sims was guilty. During this DNA analysis, many processes were used, including RFLP, SCR, PCR, as well as the CODIS program. The restriction fragment length polymorphism is the first process performed during DNA testing. This is also known as RFLP. The first step of RFLP is to separate white and red blood cells with centrifuge. The second step is to extract DNA nuclei from white blood cells by bathing cells in hot water and then adding salt and then putting back the mixture in the centrifuge. The third step is to cut DNA into fragments using a restriction enzyme. The fourth step is to apply agarose gel on DNA. The fifth step is to separate and spread DNA segments by length using electric current, which is agarose gel electrophoresis. The final step is to spot DNA using a sheet of nitrocellulose or nylon. The second DNA process is STR and PCR, which stand for short tandem repeats and polymerase chain reaction. STRs are short sequences of DNA, length of 2 to 5 base pairs repeated numerous times. When using STR, if there is not enough DNA to be tested, PCR can be used to replicate this DNA. PCR allows us to recreate millions of copies of a certain DNA strand. PCR is based on using the ability of DNA polymerase to synthesize new strands of DNA complementary to offered template strand. Advantages of STR over RFLP. Although RFLP can be used to identify people and estimate the relationships between each other, STRs are much easier and are worth the payment. In RFLP, you have no way of knowing where there might have been a mutation in one of these sites, so you have to do a lot of guesswork and look at more places to find something useful. However, in STR, you know where the sites are before you begin the process. You know there are going to be some individual disparity at least some of the sites, and if you look at more than one site, you will be 99% sure of the end results. And finally, SCR takes less work and is more statistically dependable. The next thing that I'm going to introduce is the CODIS program. CODIS is made for analyzing RFLP profiles and their storage in a searchable database. Today we use it to store and search STR profiles. CODIS was created by the FBI in 1990. This program has many classifications including DNA profile entry and management, searching for profiles in the database, match management, which is managing search results, and statistical calculations, which allows labs to calculate profile statistics based on the lab or FBI's own population frequency data. Of course, in any program, there are some risks. CODIS's risks are that the DNA samples aren't always complete, and also that some agencies may hack CODIS without the person's consent. Lastly, CODIS may take an innocent person's DNA that has been found at a crime scene and mistake it for a suspect's. Going back to Randolph's case, he was exonerated last year using DNA testing and the CODIS DNA database. This may explain why there has not been any information about his life today. The only thing we know is that he is living with his daughter and son until he can get back on his feet. Thank you for listening to my podcast.